Today we are getting technical. We're diving deep into Chapter 4, Growth, Survival, and Death of Microorganisms. Now, I know what you're thinking, Chapter 4, that sounds a bit like homework. But trust me, this is where the rubber meets the road.
It really is. This isn't just about watching bacteria multiply in a petri dish. I mean, this chapter, it covers the fundamental mathematics of life, the strategies organisms use to survive in really hostile environments, and this is crucial for anyone in medicine or public health, the actual science of how we kill them.
Yeah, we're going to unpack the physics of bacterial growth. I mean, literally the equations that tell you how fast an infection can spread.
Right.
We're going to talk about the specific chemistry of sterilization, and we are going to get into some really sci-fi sounding concepts, things like zombie bacteria and cellular telecommunication.
Our mission today is to essentially explain every line of this source material. We want to take these, kind of dry charts and chemical formulas and translate them into a really clear picture of the microbial battlefield.
So let's start with that big picture. The text opens with a bit of a reality check. It's survival in the wild versus survival in the lab.
Right. And this is so important. Most of what we know, or what we think we know about bacteria, comes from studying them in these pure cultures in a lab. You give them the perfect temperature, endless food, no predators. It's a life of luxury.
It's the all-inclusive resort of the microbial world, endless buffets, perfect weather.
No responsibilities, exactly. But the source material makes a very, very strong point right at the beginning. Nature is not a resort. It is a battlefield. Out in the wild, the population of microorganisms stays, roughly constant. And why is that? Because growth is constantly being balanced by death. It's a zero-sum game.
The text brings up the concept of the microbial niche. Let's unpack that, because niche is a word we use a lot in, say, business or marketing, but it means something very specific here.
A niche is essentially the specific environmental role and the physical space an organism occupies. Think of it like a job opening in a very crowded city. In nature, there's just this intense competition for nutrients. If there's a vacant niche, say a patch of soil with a specific nutrient or a part of your gut, it will not stay vacant for long.
Nature abhors a vacuum.
Correct. It'll be filled by a consortium of microorganisms. It's
Right.
almost never just one guy. It's a team or sometimes, a gang that takes over that space.
And this brings up the human element almost immediately. The text mentions the microbiota.
Yes, the consortium of all the different microorganisms that inhabit human cells. We often think of ourselves as individuals, but we're really just walking ecosystems. And the key insight here is about physiologic efficiency. In the wild, survival isn't just about finding food. It's about the balance between nutrient availability and the energy cost of actually processing it.
So it's an economic problem. It's a return on investment calculation for a bacterium.
It is entirely an ROI problem. If you
Right.
spend more energy making the enzymes to digest a nutrient than you actually get out of that nutrient in, caloric energy, you die. The species that survive out there in the wild are the ones that have optimized their metabolic engines for that specific niche. They are efficiency machines.
Okay, so that's survival. But before we get to death, we have to define growth. And the source material is very, very specific here. It says growth is not just, getting fatter.
That's a crucial distinction. In biology, especially in microbiology, we have to be precise. The text says, and I'm quoting here, "The increase in size that results when a cell takes up water or deposits lipid or polysaccharide is not true growth."
So if a bacterium just swells up with water, that doesn't count. It's just bloating.
Exactly. It's just bloating. True growth is defined as the orderly increase in the sum of all the components of an organism. It means you are replicating your DNA, you're making new proteins, new cell wall material, and ultimately for bacteria, this all leads to one thing: binary fission.
Binary fission, one cell splits into two.
Correct. And because bacteria are single-celled, growth almost always refers to an increase in the number of bacteria in what we call a culture, rather than just an increase in the size of one individual cell.
Now, this leads us to a really practical problem. If these things are invisible to the naked eye, how do scientists actually measure their growth? The text outlines 3 different methods, and they seem to be measuring slightly different things. And honestly, this part confused me at first. If I want to count them, why can't I just stick them under a microscope and count? Why do we need these 3 complex methods?
You could try, but imagine trying to count 1,000,000 tiny marbles floating around in a murky swimming pool. It's incredibly difficult to be accurate, and it's so time-consuming. Plus, you can't easily tell the live ones from the dead ones just by looking at them under a standard microscope. So we need proxies. We need better ways to estimate.
Okay, let's look at the options. The first distinction the text makes is between cell concentration and biomass density.
Right. Cell concentration is the number of viable cells per unit volume. So how many individual living bacteria are in, say, this 1 milliliter of fluid? Biomass density, on the other hand, is the dry weight of all the cells per unit volume.
And the text says these two things aren't always the same. Why not?
Because the average dry weight of a cell can change depending on what it's doing. A cell that's just getting ready to divide might be synthesizing a ton of new protein and DNA, making it heavier than a cell that's just kind of hanging out in a dormant state. So if you're studying something like genetics or, how to kill them, you really care about the number of individual cells, that cell concentration. But if you're studying biochemistry, how much food they're eating, then biomass density is the more important number.
Okay, so let's get into the methods. Method A is the viable cell count, which is also known as the plate method. This one feels like the classic high school biology experiment.
It is. It's the gold standard for many, many applications. The whole concept relies on this invisible to visible principle. You take your bacterial soup, but you can't possibly count billions of cells, so you have to dilute it.
The text mentions 10-fold serial dilutions. Can you walk us through what that actually looks like on a lab bench?
Sure. Imagine you have a test tube with your original culture. It's just teeming with billions of bacteria. You take 1 milliliter of that and you put it into a second tube that has 9 milliliters of sterile water. That's a 1 in 10 dilution. You mix it up really well. Then you take 1 milliliter from that tube and put it into another 9 milliliter tube. Now you're at 1 in 100.
And you just keep going. 1 in 1,000.
Yeah.
1 in 10,000.
Exactly. You might do this 6 or 7 times. By the end, you have a tube where the bacteria are sparse enough that you can actually count them. Then you take a tiny amount, an aliquot, and you spread it onto an agar plate, which is like a petri dish full of jelly-like food.
And the idea is that one single invisible bacterium will land on that jelly. It'll start dividing, and eventually it'll form a visible mound that we call a colony.
Exactly, a colony-forming unit, or a CFU. And there's a statistical sweet spot that the text highlights, which is really important. Scientists only count plates that have between 30 and 300 colonies.
Why that specific range? Why not count a plate that has, say, 500 colonies?
Well, if you have fewer than 30, the statistical error is just too high. It's like flipping a coin 3 times and concluding it always lands on heads. It's not a big enough sample size. But if you have more than 300, the colonies start growing into each other. Or, and this is crucial, they compete for nutrients on the plate, and some of them don't grow at all. So you get an undercount because the plate is just too overcrowded.
And the formula they give is plate count times the dilution times 10 equals CFU per milliliter.
Right. So imagine you count, say, 50 little spots on the plate, but you had to dilute that sample 10,000 times to get to that number. So you have to multiply that 50 back up by 10,000 to figure out what was in the original concentrated tube.
That makes perfect sense, but there's a really big limitation mentioned here. Dead bacteria don't count.
Correct. If a bacterium is dead, it can't form a colony. The plate just stays empty where it landed.
Which is great if you only want to know what's alive. But what if you need to know the total load, dead or alive? That brings us to method B, turbidity. The cloudiness method.
This is a much, much faster method. You use a device, usually it's a spectrophotometer, to shine a beam of light through your tube of liquid culture. The more bacteria there are, the more they scatter that light, and the cloudier or more turbid the fluid becomes.
The text gives some examples of what this looks like. It says, a barely turbid suspension of E. coli has about 10 to the power of 7 cells per milliliter.
That's 10 million cells per milliliter, and it just barely looks cloudy. I mean, it looks like maybe someone put a single drop of milk in a big glass of water. A fairly turbid suspension is 10 to the 8th. That's 100 million cells. Now that looks properly milky.
But you can't just look at it with your eye and say, that's 100 million.
Oh no, you need a standard curve. You have to first correlate that cloudiness measurement, that turbidity reading, to a known viable count that you did earlier using the plate method. But here's the big catch with turbidity. It measures everything. Live cells, dead cells, even just little bits of cell debris if they're large enough to scatter the light.
So if you kill all the bacteria, but you don't actually blow them up, if their little bodies are still just floating in there, the turbidity stays high.
Precisely. You could have a test tube full of completely dead bacteria that looks just as cloudy as a tube of living ones, and that's a really crucial nuance if you're testing, say, a disinfectant that kills without lysis, without actually bursting the cell.
Then we have method C, biomass density. This seems like the most direct method, but also the most annoying one to do.
It is definitely cumbersome. You take your culture, you spin it down in a centrifuge to get a little pellet of cells at the bottom. You wash it with distilled water to get rid of all the salt and leftover food. You dry it out in an oven, and then you weigh it. It gives you the dry weight.
It sounds a little bit like baking.
It is, yeah, but on a microscopic scale.
Yeah.
And because it's so slow, investigators usually, again, they make a standard curve. They'll do the weighing process once to figure out that, okay, this much cloudiness equals this much weight, and then they just use the fast turbidity method from then on.
Okay, so we know how to measure them. Now let's talk about how they actually grow. This is where the math starts to kick in. Section 2, the mathematics of exponential growth.
This is really the engine of the entire microbial world. We mentioned binary fission before. 1 cell becomes 2, 2 become 4, 4 become 8. It's a geometric progression.
The text gives us a breakdown of the formula on page 56. It says that the rate of growth is first order. What does that mean in plain English? Because in physics or chemistry, first order has a very specific meaning.
Right. First order kinetics means that the rate of the reaction depends directly on the amount of substance you have. So in a factory, if you have one assembly line making cars, adding more raw steel to the warehouse doesn't make that line go any faster. The rate is fixed. But in biology, the product is the factory.
Oh, I really like that. The product is the factory.
Exactly. Every time you make a new bacterium, you have also just made a new machine that can make more bacteria. So the more cells you have, the faster the population grows in total numbers. The equation is dBdt equals kBb.
Okay, let's translate that for everyone.
Okay, so dBdt is the speed of growth. It's the change in biomass over the change in time. B is the current amount of biomass you have, and K is the growth rate constant.
Let's focus on that K. This seems like a really important number for comparing different kinds of bacteria.
It is. K represents the intrinsic rate at which that specific organism produces more cells under those specific conditions. It's like the horsepower of its engine. The text gives a really striking comparison. Some rapid growers have a K of 4.3 per hour.
4.3 per hour.
That means for every 1 gram of cells you have, it produces 4.3 grams of new cells every single hour. That is just explosive growth.
And the slow growers?
Some of them might be as low as 0.02 per hour.
That is a massive, massive difference.
It's huge. And to help visualize this, the text talks about equation 3. If you plot the biomass against time on a normal piece of graph paper, you get a curve that just shoots up like a rocket. That's your classic exponential curve. But scientists, well, they kind of hate curved lines because they're hard to extrapolate. So if you plot it on a logarithmic scale, you get a perfect straight line.
And the fact that it becomes a straight line on a log scale is basically the proof that it's exponential growth.
Correct. It's the hallmark of it.
Now, there are two terms here that sound almost the same, but the text says we should distinguish them. Doubling time, which is TOD, and generation time, TOG.
Practically speaking, they are usually the same thing. It's the time it takes for the population to double in size. However, generation time specifically implies the time for one single cell to divide and become 2. Doubling time is the time for the total biomass of the culture to double.
And when would they not be the same?
Imagine a situation where cells are getting larger, but they haven't divided yet. The biomass might double before the actual cell count doubles. Or, in more complex organisms like fungi that grow as these long filaments, generation time becomes a really fuzzy concept, but the doubling time of the total mass is still very measurable.
And there's a formula that connects them to K. K equals the natural log of 2 divided by the doubling time. Or roughly 0.693 divided by TD.
Right. So if you know how fast they double, you can easily find K. If you have a doubling time of, say, 10 minutes, which is incredibly fast, by the way, that's 0.17 hours, you plug that in and you get that massive K value of 4.1. If the doubling time is 35 hours, K drops all the way down to 0.02.
And here's where it gets really interesting for me, using this math to predict the future. The text uses these formulas to calculate how long it takes for a culture to reach a certain size.
Yes, this is the slow grower example.
Yep.
Imagine a pathogen with that really low K of 0.02. It grows very, very slowly. If you start with just one single cell and you want to know how long it'll take to reach 107 cells, which is that barely cloudy suspension we talked about, you can just plug it into the equation.
You know, what's the answer?
It's about 800 hours.
That's over a month.
Over a month. Now, compare that to something like E. coli, which could do that in a single day, easily.
So the text makes a kind of philosophical point here. It says, the race isn't always to the swift.
This is such a key evolutionary insight. You would think that the fast grower always wins. But if you're a slow grower, you might survive because you can metabolize some nutrient that the fast grower can't even eat. Or maybe your slow metabolism makes you naturally resistant to antibiotics that specifically target rapidly dividing cells.
It's the difference between a sprinter and a long-distance hiker. The hiker might take a lot longer to get there, but they might be the only one who can actually handle the rough terrain.
Exactly. Or, while the fast grower triggers the immune system immediately and gets wiped out, the slow grower can fly under the radar, establishing a chronic infection before the body even knows it's there. Tuberculosis is a classic slow grower. It plays the long game.
So far, we've been talking about growth as if it just goes on forever. But we live in a finite world.
Yeah.
This brings us to section 3, the growth curve in a batch culture.
Batch culture is a really crucial term here. It just means a closed system. Think of a test tube or a flask. You put the food in, you put the bacteria in, and you seal it up. No new food comes in and no waste goes out.
It's a sealed room.
It's a sealed room. And in this sealed room, we see a very predictable drama unfold in 4 acts. And this is what we see in figure 4.2.
Act 1, the lag phase.
You inoculate the bacteria into the fresh medium, and nothing happens. The line on the graph is just flat.
So they're just dormant.
Yeah.
Taking a nap.
That is the big misconception. They are absolutely not dormant. They are frantically retooling. I mean, imagine you just woke up in a completely new environment with totally different food sources available. You need to build the tools, the enzymes to digest this specific food. You need to replicate your DNA. You need to increase your cell mass.
The text mentions something called the inoculum effect. This sounds almost like a socioeconomic problem for bacteria.
It kind of is. If you take cells from a very rich medium where they had everything handed to them like a trust fund environment, and you drop them into a poor medium where they have to synthesize their own amino acids, the lag phase is very long. They have to downshift and start synthesizing all the machinery they used to just get for free.
They have to learn how to cook for themselves.
Exactly. But if you take them from an exponential phase culture and you put them right back into the exact same medium, the lag phase might be 0. They just keep on going.
Okay, so then we hit Act 2, the exponential phase, or the log phase.
This is the boom times. The retooling is done. The cells are dividing at that maximum rate, that K we talked about. The graph shoots up in a straight line, at least on a log scale. New cell material is being synthesized at a constant rate.
But in a closed system, in that sealed room, the party can't last forever.
No, it can't. Eventually, one of 2 things happens. Either the food runs out or the guests trash the house.
Toxic byproducts.
Exactly. Bacteria produce waste things like organic acids, other toxins. In a closed tube, that waste just builds up and up, so either starvation or toxicity is going to hit.
Which leads us to Act 3, the stationary phase, the plateau.
The curve flattens out. But again, this isn't a pause. It's what we call a dynamic equilibrium. We see something called cell turnover.
And what does that mean? Cell turnover.
It means that cells are dying and new cells are being born at roughly the same rate. So the total cell count might still increase slowly because the dead bodies are still floating around. But the viable count, the number of living cells, stays constant. It's a state of 0 net growth.
And finally, Act 4, the death phase.
The decline. The toxicity becomes too much, or the energy reserves are just completely gone. The viability starts to decrease. And interestingly, the text notes that death, just like growth, is exponential. It's a defined rate of decline.
But there's a tail at the end of this curve. This part sounded a bit gruesome in the notes.
It is fascinating. The death rate often decreases drastically after the vast majority of cells die. You're left with a small number of really hardened survivors, and they persist for months, sometimes by cannibalizing the nutrients that are released by their dead siblings.
Cannibalism as a survival strategy.
Well, when you're a single cell in a test tube, you do what you have to do. The lysis, the bursting of the dead cells, releases amino acids and carbon that the few remaining survivors can use to just maintain themselves.
Now, this batch culture, this whole closed system, is how we usually study them. But Section 4 talks about some advanced concepts that act really differently. Specifically, The chemostat.
The chemostat is basically the opposite of a batch culture. It's an open system. You have a vessel, and it has an overflow siphon. You are constantly dripping fresh medium in, and the overflow siphon is removing the old medium and some of the cells at the exact same rate.
So we've just described the life cycle of bacteria in a tube. It's a tragedy in 4 acts. But that's not how we work, is it? Our bodies aren't sealed tubes. We eat, we use the bathroom, we're constantly refreshing.
Exactly. We are continuous flow systems. The chemostat is designed to model this. By constantly adding fresh food and removing waste, you can achieve maintenance of the exponential phase. You can keep the bacteria young and growing forever. You never hit that stationary phase because the nutrients never run out and the toxins never build up.
That sounds like the fountain of youth for bacteria.
It is, and it's incredibly important for industrial microbiology. If you're using bacteria to make something like insulin or an antibiotic, you don't want them to get old and stop working. You want to keep them in that high-energy, productive, exponential phase permanently.
Now, another advanced concept that completely changes the game is biofilms. The text says that bacteria aren't usually loners.
Right. We tend to study them as planktonic, just free-floating swimmers in a broth. But in the real world, in your mouth, on a river rock, inside a medical catheter, they are sessile. They settle down and they form a community called a biofilm.
So the plaque on my teeth is a biofilm.
Yes, that is a classic biofilm. And the slime in water pipes, the process is really fascinating. A single bacterium attaches to a surface. It divides. But instead of the daughters floating away, they stay attached. And they start to produce this sticky matrix called the glycocalyx. It's basically a sugar slime fortress.
The text calls it a telecommunication system. This is the sci-fi part I was waiting for.
This is quorum sensing. It is absolutely mind-blowing. These bacteria, they produce these small molecules, things like homoserine lactones, and these molecules just float around. When the population density gets high enough, the concentration of these molecules hits a certain threshold.
And they can hear each other.
Essentially, yes. It's like being in a big room where everyone is whispering the word attack. At first, you can't hear anything, but as more and more people enter the room and start whispering, the sound builds. When that chemical signal gets loud enough, it triggers a genetic switch in all the bacteria at the same time. They turn on specific genes as a group.
So they start acting like a single multicellular organism.
Precisely. They might all turn on virulence factors to attack the host, or they might all ramp up slime production. And this biofilm structure gives them a massive strategic advantage. The cells on the very outside of the film, they get all the oxygen and nutrients, but they also face the antibiotics and the immune cells. They're the shield.
And the ones on the inside.
The cells deep on the inside are starving and they're dormant. But because they are dormant, antibiotics often don't kill them, since many antibiotics only kill actively growing cells. And the slime itself prevents immune cells from ever reaching them.
So you can kill the outer layer, think you've won, you stop the treatment.
And the inner layer wakes up, eats the dead outer layer for food, and the infection comes roaring back. This is why biofilm infections on implants, like artificial hips or heart valves, are so devastating. Often, the only way to cure them is to surgically remove the entire implant. You can't just drug it away.
Speaking of things that are hard to kill, let's talk about the zombie state. The text calls it viable, but not culturable, or VBNC.
This is a complete nightmare for public health. Sometimes when bacteria face starvation or some other stress, they enter this deep dormant state. If you try to plate them out on an agar plate, method A from earlier, they won't grow.
So you look at the plate, you see 0 colonies, and you think, great, the water is safe to drink.
Exactly. You think they're dead, but they're not. They are VBNC. They're still metabolically active, but they're just paused. The real danger is that if these zombie cells are passed through an animal or if conditions suddenly improve, they can resuscitate.
They just wake up and start causing an infection all over again.
Yes. It challenges our entire definition of what is safe. It means that 0 colonies on a plate doesn't always mean 0 risk.
Which is a perfect segue to Section 5, the definition and measurement of death. This seems almost philosophical. What actually is death for a microbe? I mean, it doesn't have a heartbeat to stop.
Right. It's not a cessation of heartbeats or brainwaves. The microbial definition of death is the irreversible loss of the ability to reproduce.
Even if it's still wiggling around.
Even if it's still metabolizing a little bit of sugar, if it cannot divide, it is genetically dead. It's an evolutionary dead end. The empirical test for this is, does it form a colony on a suitable medium?
Wait a minute, you just said that VBNC cells don't form colonies, but they aren't dead. This feels like a contradiction.
And that is the huge caveat. The conditions of the test matter immensely. The text gives a really specific example ingesting UV light. So imagine you have a culture of bacteria. You hit them with a blast of UV light. This damages their DNA. Now, if you take those cells and you immediately plate them on a salty, kind of harsh agar plate, they won't grow. You'd count them as dead. You'd say, wow, 99.9% killed. But what if you take those exact same irradiated cells and you put them in a gentle, nutrient-rich broth for just 20 minutes first, a little recovery period, before you plate them?
They repair their DNA.
They use that 20 minutes to frantically stitch their DNA back together.
Yeah.
Then you plate them, and they grow just fine.
So, were they dead in the first experiment or not?
It's Schrodinger's bacterium. In the first experiment, they were dead by our operational definition. In the second, they were alive. It means that death is defined by the conditions of recovery. And this has huge implications for testing disinfectants. If your test is too harsh, you might overestimate how good your cleaner actually is.
Now let's look at the math of death. Sterilization kinetics.
Right. Just as growth is exponential, death is usually a logarithmic decline.
Meaning?
Meaning you don't kill a fixed number of bacteria per minute, you kill a fixed percentage of what's left.
So if I have 1 million bacteria and my disinfectant kills 90% in 1 minute.
You have 100,000 left.
And in the next minute.
It kills 90% of the survivors. So now you have 10,000 left. Then 1,000. Then 100.
It never technically hits zero mathematically?
Mathematically, it's an asymptote. It gets closer and closer to zero, but it never quite touches it. Practically, of course, eventually you get to less than 1 cell, which means zero. But this is why 99.9% effective sanitizers can still leave plenty of bacteria behind if you start with 1 billion of them. If you have 1 billion, 99.9% killed, still leaves 1 million survivors.
The text shows figure 4.3 with 2 different kinds of death curves. Curve A and curve B.
Curve A is what we call the single-hit curve. It's a perfectly straight line on that log scale. This implies that there is a single critical target in the cell, say a single chromosome in a haploid organism. If you hit it once, the cell dies. It's a sniper shot.
And curve B.
Curve B has a shoulder at the start. The line stays flat for a little bit before it starts to drop. This is the multi-hit curve. It implies the cell has multiple copies of the target. Maybe it has 4 copies of a crucial gene. You have to destroy all 4 copies to kill the cell.
So that shoulder at the beginning is the time you're spending destroying the backup copies.
Exactly. You're just chipping away at their armor. Once you finally get to the last copy, the curve drops just like the single-hit version.
This brings us to the war we wage against them. Section 6. Environmental control and terms. We throw around words like sanitize and disinfect all the time, but the text defines them very rigorously in Table 4.3.
In microbiology, you really cannot mix these up. Sterilization is the absolute peak. It means the process destroys all forms of microbial life, and that includes bacterial spores.
And spores are the hardest thing to kill.
By far. Spores, specifically endospores, are the bunker mode of bacteria. They can survive boiling water, radiation, harsh chemicals, you name it. If a process can kill spores, it's sterilization. If it can't, it isn't.
And what is disinfection?
Disinfection eliminates pathogens, the bad guys, but it does not necessarily kill all the spores.
So a surgeon needs sterilization for their scalpel. A janitor needs disinfection for the floor.
Generally, that's a good way to think about it, yes. Then you have pasteurization. This is specifically applying heat, usually to milk or cheese, to kill specific pathogens like salmonella or listeria without boiling the food and ruining the taste. It does not sterilize. Milk still goes bad eventually because the spoilage bacteria can survive.
Sanitization.
That's a public health term. It just means reducing the microbial levels to a safe limit that's defined by law. It doesn't mean zero. It just means clean enough that you probably won't get sick.
And just plain cleaning.
Cleaning is just the physical removal of visible soil, washing dirt off a potato. It removes microbes physically, but it doesn't necessarily kill them.
The text also makes a big deal about distinguishing between ocidal and cystatic.
Oh, this is a crucial distinction. Bactericidal means it kills. The damage is irreversible. Bacteriostatic means it just inhibits growth. It pauses them.
So if I use a bacteriostatic agent and then I wash it away,
they'll start growing again. And this is really important in the world of antibiotics. If you give a patient a bacteriostatic drug, you are relying on their own immune system to actually come in and finish the job while the drug holds the bacteria down. If the patient has no immune system, a static drug might not work.
That makes a lot of sense. Okay, now let's get into the how, Section 7. Mechanisms of biocide action. How do we actually kill these things at a molecular level? We have a few key strategies here. Strategy 1, destroy the wall.
Right. Mechanism 1 is disruption of the cell membrane or the cell wall. The text uses this great analogy here, the corseting structure.
I love that image. It's so vivid.
It is. Imagine the cell wall as this tight fishing net that wraps around the cell. The cell inside has very high internal pressure, osmotic pressure. It wants to expand. That net is the only thing keeping it from exploding.
So if you cut the net,
The cell bursts. Lysis. Agents like penicillin or enzymes like lysozyme, that's how they work. They just snip the threads of that net. Or you can attack the membrane itself, the delicate skin under the net. If you poke holes in that with alcohol or detergents, the cell leaks out all its vital fluids or toxins flood in. It's like popping a water balloon.
strategy 2, scramble the machinery. Protein denaturation.
Proteins are these complicated, folded 3D shapes. They need that specific origami shape to do their job. Things like heat, alcohol, or acids cause those proteins to just unfold.
Like cooking an egg.
Exactly like cooking an egg. When you fry an egg, the clear liquid turns into a white solid. That is protein denaturation. And crucially, you can't uncook an egg. Once the cell's enzymes are cooked, the machinery is just fused and broken. The cell is dead.
Strategy 3, handcuffs. Disruption of free self-hydryl groups. This sounds very, very chemical.
It is. Many enzymes have an amino acid called cysteine, and it has this sulfur hydrogen or SH tail. Think of these little sulfur tails as the hands that the enzyme uses to grab onto molecules and do its work.
So what happens to them?
While heavy metals like mercury or silver or oxidizing agents like chlorine, they come in and they bind to that sulfur. They essentially handcuff the enzyme, or they'll tie its two hands together. The enzyme is still there, but it can't move. It's locked up.
Strategy 4, nuclear warfare. Damage to the DNA.
This is radiation. UV light specifically causes something called thymine dimers.
What's the dimer?
So DNA is made of letters A, C, T, and G. If you have two T's or thymines right next to each other on the strand, UV light can hit them and make them fuse together. It creates a physical bump on the DNA strand.
So when the cell tries to read its own DNA instruction,
It hits the bump and it derails like a train. It can't replicate. It blocks the tracks.
and ionizing radiation like X-rays or gamma rays.
That is even more violent. That just smashes the DNA backbone itself. It literally snaps the double helix in half. It's like taking a pair of scissors to a film reel.
Finally, strategy 5, the spy game. Chemical antagonism.
This is competitive inhibition. You introduce a chemical that looks almost exactly like the food that the bacteria needs to survive.
An imposter.
A total imposter. The enzyme goes to grab its food, but it grabs the imposter instead because it fits into the lock. But the imposter doesn't work. It just sits there, blocking the enzyme from grabbing the real nutrient.
The text mentions carbon monoxide versus oxygen as a classic example of this.
Right. Carbon monoxide binds to your hemoglobin way, way tighter than oxygen does. It takes the seat and it just refuses to move. Sulfa drugs work this way too. They mimic a vitamin that bacteria need to make folic acid. The bacteria grab the drug, try to use it, fail, and then they starve.
So we have the strategies. Now let's look at the actual arsenal in Section 8. The text lists a lot of specific agents. I feel like we usually just gloss over this stuff. But let's actually dig into these chemicals because we use them every single day.
We really do. And we can group them into physical methods and chemical methods.
Okay, physical first. Heat.
Heat is king. It is the most reliable way to kill microbes. But the text makes a really big distinction between moist heat and dry heat.
And moist heat is better. Why is that?
Water carries heat energy much more efficiently than air does. That's why you can put your hand in a 200-degree oven for a second and be fine. But if you put your hand in 200-degree boiling water, you are instantly and severely burned. Steam penetrates into the cells and cooks them much faster.
So that's why hospitals use autoclaves.
Yes. An autoclave is basically a giant high-tech pressure cooker. It uses steam under pressure 15 pounds per square inch to raise the temperature to 121 degrees Celsius. And that is the magic number. 15 to 20 minutes at 121 degrees C kills everything, including the spores.
Okay, moving to the chemicals. The biocides. Let's run through the table. Alcohols. Ethanol and isopropanol. This is what's in hand sanitizer.
Classic disinfectants. But here's the surprise that always catches people. 100% alcohol is less effective than 70% alcohol.
That feels completely counterintuitive. I would assume the pure stuff is stronger.
It's for two reasons. One, alcohol actually needs water to denature proteins effectively. The water helps the alcohol penetrate the cell wall. If you just use 100% alcohol, it coagulates the proteins on the outside of the cell wall instantly, and it forms this protective crust that actually shields the inside of the cell.
So it seals the fortress from the outside.
Exactly. The bacteria survive inside. 70% alcohol acts a little bit slower, it penetrates deeper, and it kills the whole cell from the inside out. Also, pure alcohol just evaporates too fast. You need what we call contact time.
Aldehydes, glutaraldehyde and formaldehyde.
Now, these are the heady hitters. They are alkylating agents. They can achieve full sterilization. They kill spores. But they're very toxic, and they're carcinogenic. You use these for medical devices that you can't put in an oven, like a fiber optic endoscope. You have to soak it in glutaraldehyde.
Halogens, chlorine and iodine.
Chlorine is your basic bleach. It's an oxidizing agent. When you put bleach in water, it forms hypochlorous acid, or HOCl. And this is just a potent oxidizer. It literally burns up the cell components chemically. Iodine is similar, and it's often used as what we call iodophores for a slow release on skin before surgery. It's what stains your skin that orange color.
Heavy metals, silver and mercury.
We don't really use mercury much anymore because of the toxicity. Remember the old mercurochrome? That's long gone. But silver is making a huge comeback.
Where do we see silver now?
In burn creams, silver sulfatazine. It prevents infection and really bad open wounds. You also see silver nanoparticles woven into athletic wear to stop odors. The silver ions go in and they bind up those sulfhydryl groups we talked about earlier, the granddaddy of them all. Joseph Lister used phenol to revolutionize surgery. It works by disrupting membranes. It's that classic clean smell in many hospitals. But pure phenol is very harsh on skin, so now we use derivatives, like the stuff you find in Lysol.
And finally, quats, quaternary ammonium compounds.
These are cationic detergents. They have a positive charge, and bacteria generally have a negative charge on their surface.
Oh, they attract each other.
Like magnets. The quat sticks to the bacterium, it disrupts the membrane, and maybe causes some leakage. They're great for cleaning floors and surfaces, but they are generally not sporicidal. They won't kill the really tough stuff like C. diff spores or tuberculosis very easily.
Now, using these weapons requires some strategy. Section 9 discusses the kinetics of killing, specifically the relationship between concentration and time. This is the part that kind of terrified me about how I clean my kitchen.
This is probably the most practical takeaway for anyone listening. The formula is C to the power of N times T equals K.
Okay, so C is concentration, TID is time. What is N?
N is the concentration exponent, and it measures how sensitive the killing time is to dilution.
The text gives a pretty shocking example for phenol, where N equals 6. Let's do the math on this out loud because N6 sounds abstract until you actually run the numbers.
Okay, so let's say you have a concentration of 1. And at that concentration, it takes 10 minutes to kill the bacteria.
Okay, 10 minutes. I've got that.
Now, you decide you want to save some money, and you dilute the cleaner by half. So your new concentration is 0.5.
I would assume it just takes twice as long. 20 minutes?
That would be true if N was 1. But for phenol, N is 6. The change in the time is related to the change in concentration raised to the power of 6.
So we were looking at 2 to the power of 6.
2 times 2 times 2 times 2 times 2 times 2. That's 64.
64.
So if you dilute the phenol by half, it takes 64 times longer to work.
So 10 minutes becomes 640 minutes? That's over 10 hours.
Exactly. You just turned a 10-minute disinfectant into a 10-hour soak just by watering it down by 50%.
That is absolutely mind-blowing. So a little bit goes a long way does not apply to disinfectants.
Absolutely not. Dilution is the enemy of disinfection. If the bottle says use full strength, you use full strength. If you dilute it, you are, for all practical purposes, washing with dirty water.
Does this apply to all chemicals? Isn't always that high?
No, and varies a lot. For hydrogen peroxide, N is roughly 1. So if you dilute peroxide by half, it takes about twice as long. That's linear, that's intuitive. But for things like phenols and alcohols, N is very high. You mess with the concentration at your own peril.
Finally, can you reverse death? The text talks about reversal of action.
Well, we mentioned that bacteriostatic just pauses them, and you can reverse that effect in one of 3 ways: agent removal, substrate competition, or agent inactivation.
Agent removal is just washing it off.
Yep. If you put a static drug on some cells, then you spin them down and wash them with clean water, they'll wake right back up.
Substrate competition.
Remember the imposter strategy. If you flood the system with the real food, the enzyme will eventually, just by chance, grab the real food instead of the imposter. You just outcompete the poison with sheer numbers.
And agent inactivation. That sounds like an antidote.
It is, basically. For example, if you use mercury to bind up those sulfur groups, you can then add a compound that has lots of sulfur in it, something like thioglycolic acid.
And the mercury prefers the acid.
It creates a much stronger bond, so the mercury will let go of the enzyme to bind to the acid instead. The enzyme is freed, and the bacterium wakes up.
It's chemical warfare, but with a restart button.
It is, and it's why laboratory testing has to involve neutralizers.
Yeah.
If you want to know if a disinfectant really killed the bacteria, you have to add a neutralizer to the tube at the end to make absolutely sure they aren't just sleeping.
So what does this all mean? We've gone from the invisible universe to the math of binary fission, to zombie states, and finally to this whole chemical arsenal.
Well, if we connect this back to the bigger picture, it really changes how you see the world. I mean, we are not living on some sterile planet. We are immersed in a microbial soup that is constantly growing, adapting, and fighting for equilibrium.
In our attempts to control it, cleaning, antibiotics, sterilization, they're all governed by these very strict laws of physics and chemistry. It isn't magic. It's concentration exponents and growth constants.
And as the text reminded us right at the beginning, the race is not always to the swift. The bacteria that grow slowly, that hide in those biofilms, or that enter that VBNC zombie state, those are the ones that often outsmart us in the end.
That is a very humbling thought. Next time you wipe down your kitchen counter, think about that next exponent. You're not just cleaning. You're engaging in high-stakes chemical warfare.
And physics is on your side, but only if you follow the rules.
That's it for this deep dive. Keep those surfaces clean and keep your mind open. We'll see you next time.